welcome. This is, I think, our 10th anniversary symposium. So, for those of you that don't know the program, people say I do. So the program started about 10 years ago between ourselves and a colleague at the University of Virginia, Zygmunt. And now it's expanded to a point in which University of Texas Southwest, UT Southwest, has joined has joined the Oklahoma Medical College. So there are 4 universities, typically Chicago and UVA take more students than the others just because they have smaller programs. And another thing that's happened over the last 2 years is the Fulbright Foundation has started to participate. So they're very active and a very important part of the program. So this year we have 8 students over the years. I think we have about 70 students that have gone through the program, and some stick around, some we can't get rid of, and those go to place back in Europe and so forth, getting very successful now. So what happens in this, at least in terms of determining the students that become and participate in the program, it's a very, it's a difficult program to get into. So there's a set of criteria or metrics that have to be, each student has to have a certain grade point average and a certain background, and that's vetted now by the Fulbright Commission. So by the time my colleagues and myself get to Poland, they're been triaged down to about, this year we had 55 interviews in 3 days. So you can imagine that's a lot of work. And so everybody that's part of the program has a very good academic record and a lot of enthusiasm. That's part of the interview process. Is we look for students that not only have accomplishment, but also try to pick people that have a lot of, that's what some people have done pretty well doing. So we have a full program today, and I think the presentations will be 15 minutes and 5 minutes questions. I urge you to ask questions. That's part of the deal. And so again, look forward to it. Lanny, could you come up and introduce our first speaker? Why am I the only person in the front row up here?
So I'm so pleased to introduce to you Tomasz Szlebda. Yeah, so Tomek really struck me with like universal gifts. He has gifts everywhere. It's like very well educated, very smart, very well organized, and on the top of all of this, he's so well mannered. It's such a pleasure to deal with him. Thanks to your parents. And
he's also my dog sitter too. One of my dog sitters.
So, and he doesn't like to just waste time. He is a great researcher. He's very, very dedicated a student. He spends most of his time in the lab, but on the top of everything, he likes to travel. And he even been to Cuba this year. And this is always was my great dream to go there. So I hope I will follow you there sometime. So as to his work, he has done so many different projects. Probably you know that we have the CBC collaboration going on in the lab, and within this collaboration we got orders to produce, to generate the synthetic antibodies for different antigens. And Tomek this year, I don't remember, maybe 5, 6 projects he has done just within the CBC. But his main project was Tony decided to use different parts which was generated in the lab through the years and put them all together. He asked Thomas to use the newly engineered variant protein G1, and selected fabs for Ebola nucleoprotein and mix them together and make the universal model detection assay in order to be able to detect many different antigens. And Tommy did it just perfect. Honestly, I didn't have high hopes when we began. And it's not
First good impression, I know.
yeah. But it worked. And here he is, and I cannot be more happy.
Thank you so much, Lena, for introduction. I blush, I can't say. And yeah, you can see that. I'm not happy that I didn't make the great first impression, but I'm happy that I followed later. So I would like to tell you more about my main project, as Elena mentioned, which was to develop a modular universal detection assay for multiple targets, and how we try to apply this detection assay for Ebola virus detection. In 2014, Ebola outbreak in West Africa showed us the urgent need for a point of care which can detect early stage on Ebola virus infection. Currently engineered detections for Ebola virus are expensive and very time consuming. What is needed in Africa and all over the world is fast, easy and inexpensive diagnostic which everyone can do, not only qualified person. That's why we decided to develop split enzyme protein G based sandwich immunoassay SEGIA. And SEGIA is using three major components. Fabs, which are monovalent part of IgGs which binds to the antigen. Those Fabs have to recognize different epitopes on the antigen. Protein G, which binds to the Fabs. And split enzyme, which are linked to the protein G. The concept is that when the parts of the split enzyme are brought close enough by two independent binding of the Fabs to the antigen, brought them close enough that molar concentration of the Fabs of the parts are high enough for refolding and reconstitution enzymatic activity which we can detect. First, I would like to tell you more about this interaction between protein G and the Fab. So, why we choose protein G? There's several bacterial proteins which binds to the Fab, like protein A binds to the heavy chain of the Fab and protein L binds to the light chain of the Fab. Both of those proteins binds to the Fab with very high affinity. But they bind very close to variable parts of the Fab where the CDRs are located. This might interfere of the Fab binding to the antigen. And there is protein G, one more protein which binds to the constant part of heavy and the light chain of the Fab. Major interaction between protein G and the Fab is through the beta strand. Major interaction of protein G is through the beta strand and heavy chain of the Fab, and here helical cap and the light chain of the Fab. Affinity of this binding is 3 micromolar. It's not good enough for detection assay. That's why we decided to use recently engineered in our lab protein GA1 variant. Affinity of this binding is 24 nanomolar, which is huge improvement, but still not good enough for detection assay which we try to develop. We had to improve this complex. To do so, we decided to maturate Fab light chain scaffold because it was observed that different IgGs have diversity at the interface of binding to protein G. Here I'm showing you the interaction between protein G and the Fab through the light chain. From this interaction, we choose those 5 amino acids to be randomized as we try to improve this binding. To do so, chemical mutagenesis have been done to produce Fab light chain scaffold libraries. As a template, we use phage meet of 2 Fabs, MG6 and MG20, about which I would like to tell you more lately. We use two different primers, where NNK covers all 20 amino acids, NNT covers all amino acids except stop codons and tryptophan. And KTG covers only two amino acids, leucine and valine. Those are the only two amino acids which were observed in all IgG isotypes in position 125. Here we can see the chemical flow, and as a result we got 4 libraries contains 10 to the 9th clones each. This is 100 times more than theoretically calculate number of variants in our libraries, and those libraries were used for phage display selection. Protein GA1 was exposed for newly generated Fab light chain scaffold libraries. After 5 rounds of selection, we got enrichment in phage specificity, and 96 clones were checked by ELISA. The best looking clones on ELISA level were sequenced. From 19 sequenced clones, we got 7 different Fab light chain scaffold variants, from where 3 different looking on ELISA level were variant with 2 spontaneous deletions and LRT scaffold. The mostly present scaffold is SMLRS, and yes, and here I'm showing you the web logo of maturated scaffold. As you can see, at position 125 we got only leucine, and in position 126 only arginine in all variants of maturated scaffold. This result means that those 2 amino acids have a critical role in Fab protein GA1 interaction. To characterize those scaffolds, SPR for selected variants have been done, and the results show that the scaffold with 2 deletions have improved affinity of binding to 1.3 nanomolar, and SMLRS, the mostly present scaffold, have 7.5 nanomolar affinity respectively. In our hands, non-treated Fab light chain scaffold have more than 100 nanomolar affinity, but as you can see on the off rate, our variants, we significantly improve the binding interaction between protein G and the Fab by improvement in off-rate kinetics. At this point, we are trying to reveal the structure of newly generated Fab light chain scaffold and protein GA1. And at this point of when we have this result, we are ready for application, which can be used in our assay. So I already told you about this interaction. Now it's the last part. Enzyme, which is splitted and inactive when splitted, and can be refold and reconstitution when 2 parts of the Fab, when two Fabs bring 2 parts close enough, so the molar concentration of the parts is high enough for refolding and reconstitution. There is several split enzyme systems, but we choose to use beta-lactamase system, which was recently published in vivo study of protein-protein interaction. This enzyme was reported as easy to split. In this study, they fused one protein with one part of the beta-lactamase called BLF1, other protein with other part of beta-lactamase called BLF2. And so we followed their beta-lactamase design. We split our enzyme in position 196, and in position 185 we introduced substitution methionine for threonine, which was observed and which was reported as important for this complex reconstitution. In this study they are using chromogenic substrate. In our assay we are using fluorescent substrate. And yeah. So at this point we're ready. We have all SEGI components. The only thing we need is a modular system from where we can estimate the reaction condition. To do so, we choose yeast ISF1 protein and their 2 Fabs, 5E and 12E, which were recently engineered in our lab. And structure of those interactions is known. We need to figure out how long our linker between protein G and part of beta-lactamase have to be. So from the structure, we can calculate that N and C termini distance is between 100 and 150 angstrom, which suit to 30 amino acids linker concept, what we've done. Here I'm showing you the structure of Fab 11E and 12E. Structure of Fab 5e is not solved yet, but we know that Fab 5e binds very close to the place where Fab 11e binds. That's why we are using this modular system. It was a time to set up real experiment with Segia. We were very excited about that. When Elena came to me after the first day and asked, "Is the result good?" my answer looked more or less like this. Yes, Segia works. With our modular system, we can see increase of the fluorescent signal when the antigen is present. What is more, we did the titer of antigen concentration, and here I'm showing you two figures which explain what is happening here. These titers show us that our result is real. At this situation, when the antigen is in low concentration, two fabs recognize epitopes on the same molecule, which result in beta-lactamase refolding. In this situation, when antigen is in high concentration, fabs recognize epitopes on different molecules, which result in decrease of the signal, right here. So, we are ready. Segia works. We know that it works in modular system. What we have to do now? We have to apply it for Ebola virus detection, which was our main goal when we are developing this assay. So, we need two fabs which recognize different epitopes on Ebola. We choose two fabs recently engineered by previous Polish student Mateusz Jaskulowski, fabs MJ6 and MJ20. Here I'm showing you the SPR, which shows that those fabs recognize different epitopes on Ebola zaire nucleoprotein. When we flow MJ20 fab, we see increase of the signal. When we again flow MJ20 fab, we don't see changes of the signal, but when we flow MJ6 fab, we see increase of the signal. That proves that those fabs recognize different epitopes on the antigen. Both of those fabs recognize Ebola zaire nucleoprotein with very high affinity. MJ6 0.7 nanomolar, MJ20 1.2 nanomolar. Those two players look like very good for our game for detection assay of Ebola zaire detection. That's why we decided to set it up, and it works. We have increase of the fluorescent signal when the Ebola zaire nucleoprotein is present, and titration shape look similar to the one observed in modular system. At this point, we need to optimize the reaction condition, and this is exactly what we are doing right now. And yeah, as a conclusion, I want to say that Segia can be used as a detection assay for multiple targets, and we hope that in future we will develop Segia into ultra-sensitive diagnostic tool, which can be used and solve world's problem. This is our goal. Yes, at this point I would like to appreciate some of you. First of all, my amazing boss Tony. Tony, thank you for this year. It was a pleasure. It is a pleasure to be part of your lab. I'll always remember two things about you. First, that you were a great boss, and the second thing, your happy face when you were sitting on Santa's lap. Also, I would like to thank all COS Lab people for a great environment. I would like to point here Elena for a whole year for sharing all knowledge and experience. I think I learned a lot in COS Lab, and I wish to grow more. Also, I would like to thank Shonat for all advices with crystallization and any other things, and Kasia for our downstairs lab, COS Lab environment. At this point, I cannot forget about Polish team. Thank you guys for all the fun we had together, and it was great to have friends like you, and I hope we will continue that. Thank you so much for your invitation. I will be happy to take your questions.
Take this picture out.
No, this picture is staying. Unicorn is winning here, so. Yes, please.
So I have a question about your affinity maturation. So
yes.
You showed that it was you had improved affinity through lowering the off rate.
Yes.
And maybe you said it and I missed it. Did you actually do the selection designed to lower the off rate, or is that just what came out?
It is what came out, but we did also off-rate selection, but the result was exactly the same. So after this selection, we did another selection, off-rate selection, and we got exactly the same variance of scaffold.
So what, taking it to the ultra-sensitive regime, what do you have to do?
We have to drop the we have to find the conditions where the signal of the beta-lactamase part by itself is lower at some point that we can detect real infection. So for now, our background without antigen signal is pretty high. It's getting much higher when we add antigen, but it's still too high for ultra-sensitive universal assay, which we try to develop. If we drop this signal, I think like 10 times, this is our goal, then we can detect even low concentration of antigen, and this is what we're trying to do.
So are you going to do this with a chromogenic substrate that you can actually see by?
For now we are using fluorescent because it's easier for optimization of the problem, but the main goal is to use chromogenic substrate, yes. To see the difference in the, because for fluorescent substrate we need special equipment, and chromogenic substrate will change the color of assay. This is our main goal, to have it easy and fast. Everyone who is not specialist can do it.
So, you know, in the, again, sort of follow up, like if you need to be an ultra-sensitive, what kinds of concentrations do you need to be able to detect? Because it looks like in the nanomolar range of detecting substrate. Is that reasonable? I have no idea what concentration these kinds of molecules are in say, blood or cellular fluid.
The problem is that in blood there's a lot of IgGs which will be bound by our protein G. So there will be a competition between our Fabs binding to the protein G. And, yeah, but we have to drop it to the nanomolar concentration, to the concentration where our parts of our enzymes still can get refolding and reconstitution, and it didn't give us this background.
There are mutations that will stop, protein gene mutations that will stop it interacting with the FC.
Yeah, cross-linking. This is like what we think to do later. To cross-link the Fabs to the protein G, so there will be no exchange, but this is future.
So in a positive patient, though, how much of this antigen is present?
In the
In a patient that was positive, so if you, how sensitive does your assay need to be?
very sensitive. This is nanomolar range.
Nanomolar?
This is nanomolar, yes. But so we have to
choose whether it's picomolar. You can actually get enough. You can get enough to do this.
It is picomolar that you're trying to get to, or is nanomolar sufficient?
No nanomolar should be sufficient.
That's why we're trying to get this.
There's a lot of virus.
Yeah, sure.
Yeah, this low nanomolar concentration that we need. That's why we're trying to improve this light chain scaffold, because 24 nanomolar of this binding is not good enough for us. So we're trying to drop to very low nanomolar concentration of every part, every piece. Okay, thank you so much.
Okay, next up is Kasia Gavika. Kasia, that's how you say it. Whoa. Kasia. So Kasia worked on a couple different projects this year. Both were centered around the basic question of how bacterial cells perceive stress and adapt to stress. Stress sort of broadly defined. So there's a sensor kinase that our group discovered a few years ago that is able to regulate cell survival in the face of multiple environmental stressors: oxidative stress, osmotic upshock, starvation, a whole variety of conditions this one particular protein mediates survival. So you can knock this gene out and you can see a loss of cell survival. But one question we ask about this particular sensor kinase is, can we select for mutants that are gain of function? So Kasia developed a screen to screen for gain of function mutants. And she got some interesting hits. And so I think this is going to provide some structural insight into how this particular class of sensor proteins work. So, Kasia.
Thank you for the introduction. thank you also all people who are involved in this project, in this program. Thank you, Tony, for introducing me here. It's like this possibility to develop my scientific. Oh, sorry, I'm so nervous. So this is my title.
Accent on the stress.
Yeah, it's stress. Bacteria are stress. Yeah, so for bacteria, it's crucial to sense the stress signal from environment because it depends on if they survival. So the natural environment fluctuates, so they have to rapidly react to prevent or repair the damages caused by stress, but they are not helpless. They have general stress response, and it's this particular system is widely conserved in all alpha proteobacteria, and in physiological condition where is no stress, all of the system components are inactivated or inhibited. But when kinase senses the stress signal, it undergoes the phosphorylation. The phosphate group is transferred to FIR, the response effector, and it's able to bind NEPAR and release alternative sigma factor, and it can bind to RNA polymerase and initiate the transcription of GSR gene. How you can see, the core component of these pathways are pretty good conserved. Only one of these components is not, it's kinase, which I'm interested. The kinases in a canonical pathway, the function of there is the same, so they have to go under autophosphorylation to typically results in gene expression. But the sensing domain is something what can divide them into three major groups. First of those is where the sensing domain is localized in periplasm. The second one, when sensing machinery is anchored in the membrane. And third one, where it's in cytoplasm. Kinase of main interest is it belongs to this first group. And it's named Phi-K. So Phi-K in previous studies was shown that it's necessary to stress survival and GSR transcription. It phosphorylates FIR, and gene of Phi-K is localized in GSR locus. So when we look in the primary sequence, we can see that this kinase is divided into two domains, into two parts. First of this periplasmic domain, which is marked by two transmembrane regions, which we can see on in hydrophobicity plot. And the second one, cytoplasmic, where it's really well conserved, especially in the dimerization histidine phosphorylates region and ATPase activity region. What we know about the sensing domain? It's less conserved. Only two cysteine are conserved with 100%, and one of these demonstrates to be important. When we mutate this particular cysteine, the kinase cannot sense the signals, and of course the histidine, which is responsible for autophosphorylation, also make this protein inactive. So this mutation inactivates this protein, but we are interested in mutation which can activate it. So in unrelated research in our lab, we found two mutants which are really interesting. First of these, delta 30, where 30 amino acids are deleted, and it causes that these mutants have really strong survival phenotype. The second one is point mutation, where A is substituted by B, and it's localized in linker between the HP domain and ATPase. So my aim was to understand the regulatory regions of phyK. As you know, kinase is typically inactive. When it senses the stress, it starts to be phosphorylated, so it is active. But we are interested in mutants which, in absence of stress, can be activated. It can be done by several ways. First of this, by initiate the stress perception, by mimic signaling stress, uninhibit kinase domain, or just increase kinase catalytic activity. To select these mutants, I used selection like this. So I based on fact that hyperactive mutants increase the GSR transcription. So when I put the promoter of GSR when I fused the promoter of GSR with gene of chloramphenicol, with gene which encoded the chloramphenicol resistance, I could make the selection that hyperactive mutants are resistant for chloramphenicol, so chloramphenicol can be used as a selection marker for this screen. Here I plot the final OD of my strain. It depends on the different chloramphenicol concentration, and you can see that strain lacking phyK is really sensitive. The wild type is intermediate sensitive, and my known mutants are pretty resistant. So I choose the highest concentration of chloramphenicol to make my screen because the difference between growth was the greatest. As you know how I make my selection, I will show you how I built my library. So I use the error-prone PCR to enter random mutation through gene of phyK. After this, I ligate it to plasmid carrion decanamycin resistant and transferred to my strain, my selection strain. I pour them on kanamycin plate to choose only these transformants who has my plasmid, and perform the real selection with chloramphenicol. And after this, also pour on the plate to select individual clones. To confirm that my chloramphenicol resistant clones are resistant to chloramphenicol because of the phyK alleles, not because of the another mutation, I performed a secondary screen. So I used, I transferred my plasmid with phyK alleles, with phyK mutants, to construct where gene of beta-galactosidase was under control of promoter of GSR. So I can use the colorimetric assay to evaluate LAX-Z activity as a proxy of GSR transcription. And here I plot the activity of wild type. You can see that it's pretty low. The known mutants, the activity is higher. Some of my new mutants are not. It doesn't increase this activity, so it does mean that it has to be another mutation. But I have a bunch of mutants which increase this activity, so it's the set which I will work with. The second question was if they are more active, if they are still indicible. So I treat them with sucrose to cause the hyperosmolarity stress, and also measure the beta-gal activity. And you can see that in green it's non-stressed culture, in black there are with osmotic stress. And you can see that all of my mutants are indicible. So we are still working with protein which is functional. The next step was analyze the sequence of phyK alleles, and here are my mutants with decreasing activity of beta-gal, and here I plotted my sequence of my protein. So here are the domain which are really important. And you can see that I have mutation through along whole protein, so it's quite good. But most of them have multiple mutation. But we can catch some single mutation, and it's really important because it does mean that this particular mutation in this region causes that it's hyperactive. We found also another mutation in this region, so it's just confirmed that it's important. We also catch the mutants which has the same mutation in the same position as our previous mutants, but here the substitution is from alanine to threonine instead of valine. Here I plotted all of my mutants and conserved residues which are found in this protein, and you can see that only one proline is overlapping, so that means that this particular proline is it's really important for the regulation, not for the functioning of the FAK. So to conclude, I will say that we identify many mutants which are overactive, so it's really good, but we need to determine the stress survival phenotype. And we type important regulatory regions. First of these is where it's this part where the first transmembrane region, in the first transmembrane region. The second one is this linker between second transmembrane and the HP domain, and third one on the linker between the HP and ATPase. So it's what I done, and I will thank you all of my lab for a great atmosphere, for being very helpful for me, especially to Areta for mentoring me and give me a lot of good words and a lot of time. Christy, which I work with her, but she's not here. Of course, Sean, for being part of such a great lab. Thank you very much. I'm waiting for questions.
So your activities are in Miller units. What is a Miller unit?
It's a ratio between the OD between the OD, which means how dense is our culture, between this and the OD, which we measure after the beta-gal assay, and beta-gal assay give us the it's colorimetric assay. So it's turned more yellow when the beta-gal is more active, whereas more beta-galactosidase. So it does mean that we measure the yellow color, so more yellow, it does mean that there is more beta-galactosidase, so it's more active. And it's the ratio.
Is it lean?
It's
More or less.
More or less.
I mean, you try to do it in constant conditions that are in a linear range.
Can't help it. So you had a number of mutations in transmembrane regions. Do these transmembrane regions, do they associate with each other, or do they have partners, or are they just single passes that are independent of anything else?
So the mutation in this transmembrane, or if the transmembrane,
Well,
the second one
you had some mutations where, whoops,
here,
one.
here, that we.
Yeah, and is that associated with this? I mean, it's a cartoon here, but are those transmembrane regions 1 and 2 associated with each other? Or they have binding partners of other?
We don't know because we only can estimate how this part is looking. We don't know anything about the structure of sensing domain, and probably this transmembrane domain is somehow correspond with this transmembrane, maybe in the membrane.
Is that highly conserved?
This second one? First one? No.
Go back to substitution.
Yeah, go back. But I think that it's not well constructed.
Whoops.
Yeah. partially. No, it's more or less its construct, but I think that this mutation which we provide, it's changed a little the conformation, so maybe it's easier to connect. We have to remember that this kinase is working probably when it's a dimer, so maybe it somehow corresponds to this another kinase. It's something what we have to explain. Okay, so thank you.
So our next speaker is Patricia Guzik. Patricia has been working in the lab, obviously, for the past year. And she grew up south of Warsaw. I'm not going to try to pronounce the name of the small town, but I guess it was probably about 40 minutes by car. And she got her bachelor's degree and master's degree from Warsaw University of Technology. And her degree is in chemical biotechnology. And when she came here, she had experience in the synthesis of immunoglycosides, and actually the use of those glycosides as inhibitors for enzymes for glycosidases. And we even tested them on melanoma cell lines and did some AFM on them. So she came to my lab with that background. And in my lab, she's been working with a research professional in the lab, Yamin Shao, and she's been aimed at trying to explore an area of biology that we know sort of a lot about. We know a lot about tRNA, but we don't know a lot about mitochondrial tRNA. So she's been investigating structure and function of mitochondrial tRNA. And her goal was to isolate Fabs using the technology we heard Thomas talk about. Isolate Fabs that binds tRNAs, and that was kind of the main thrust. But then, midway through, she discovered an unusual specificities in one of the Fabs, and that really changed the direction for where she was going to go. So she, from there, developed a nice assay that she's going to tell you about today. And I have to say that I've enjoyed having you in the lab. We've met pretty much every Monday since you started, and no offense, but after some of those initial meetings, you looked a little bit like your Mardi Gras picture. But after that, you got used to me, and then things were good from there. But I also want to thank Tony here who initiated this program, like he said, 10 years ago. He's had the energy to sustain it, and some really great people have come through this program, and actually who've also turned out to be really great graduate students at the university. So, thanks.
Okay, thank you, Joe, for such a nice introduction. I think it was a way too good. Okay. Also, good afternoon, everyone. Yeah, I will just switch on my presentation first. Okay. One more time. All right. Okay, so today I will present you the project I had the pleasure to work on with Yamin Shao, and it concerns Fab-based screening assay for mitochondrial tRNAs methyltransferase activity. And at the beginning, I would like to explain you why did we focus on mitochondrial tRNA, and why is it particularly tRNA leucine UUR. First of all, mitochondrial tRNAs, there is not much known about. There are not much structural data available about mitochondrial tRNAs, mostly because of their unique structural features which distinguish them from cytoplasmic tRNAs, which were canonical cloverleaf structure. And mitochondrial tRNAs are interesting because apart from many differences in secondary and tertiary interactions, they can drastically differ from canonical cloverleaf, like mitochondrial serine AGY. And in my talk, I'm going to focus particularly on mitochondrial tRNA leucine UUR, because its gene is literally a hotspot with many mutations causing serious mitochondrial diseases like MELAS. And what's more, it has because it has non-canonical secondary structure and low melting temperature, certain modifications are required for its correct folding and stability. And most core modifications are dehydrouridine, pseudouridinulations, taurine-containing uridine, different kinds of methylations. And the specific one I'm going to focus on for the rest of my talk is N1 methylation at G9. And this modification has particularly strong impact on proper folding of this tRNA. And it's formatted by multi-domain methyltransferase, which is a part of multi-domain tRNA modifying enzyme called MRPP. So now I will discuss shortly the folding dynamics of this tRNA, since it is critical in controlling tRNA function like ribosome assembly. And recent studies show that tRNA leucine is weakly stable, and it populates two conformations where the native state, the folded conformation, is just a bit more stable than non-native intermediate. And this gel, you can see on the right side, confirms that there is a small but consistent accumulation of misfolded state. Also, we have confirmed that methylation at G9 improves the folding significantly, and it produces more constrained native state. So the gel, like you can see, the gel for methylated form confirms it too here. So knowing that methylation at G9 is so important for proper folding and stability of this tRNA, we thought that having an antibody that could bind to this tRNA would be very useful in investigating its biological role. So now shortly about anti-RNA antibodies, and namely they are antigen binding fragments, because they comprise very powerful tool. Yeah, they comprise very powerful tool in investigating, in exploring RNA field. And chaperone-assisted crystallography is one of their application. And here you can see a piece of some previous work of Piccirilli lab, and this is the crystal structure of first Fab RNA complex solved at less than 2 angstrom resolution. Also, Fabs can probe conformation and location of tRNA. They can also identify their binders and serve as a probe for modification status. So taking into account all these purposes of Fabs, we attempt to select strong and specific binders against unmodified tRNA leucine. We use the phage platform displaying a library of synthetic Fabs. The target, tRNA, is immobilized on magnetic streptavidin beads, and here you can see how it's made. So the 3' end of tRNA is overtranscribed. It forms a tail, which is annealed to biotinylated DNA sequence. So, yeah, getting back to phage display cycle, I know that most of you is familiar with this, but I need to go through it. So after binding, the fraction unbound is washed out. The fraction bound is eluted with RNase A. Then we infect the cells, do amplification, and we proceed to another selection round. And after several rounds, we do expression and we test the binding of Fabs. So from the selection stage, we obtained several Fabs that bind to unmodified tRNA with nanomolar affinity. And here you can see their CDR sequences and KD values measured by filter binding. Also, we have performed electrophoretic mobility shift assay, and this is the gel for, here you can see the gel for FabF1, and you can see that the results are consistent with filter binding, since in particular concentration range of Fab, it shifts the RNA. But now, I'm going to tell you what makes this Fab really prominent. So we performed the EMSA and filter binding again, but this time against methylated form of tRNA leucine. And to our surprise, FabF1 appeared to be specific for unmodified tRNA, whereas it has no affinity against methylated form of it. So comparing those two gels, we could say that FabF1 is conformation specific. And this gave us an idea of developing an ELISA-based assay and use this FabF1 as a probe for detecting methyl groups. So the next slide visualizes quickly the whole assay. So here I have the biotinylated tRNA, and oops. So in the first step, I immobilize it on streptavidin precoated plate. Next, I use methyltransferase to methylate G9. And now the part of tRNA populations is methylated, the other remains unmodified. And next comes FabF1, which recognizes unmodified tRNA. It's followed by a secondary antibody, which is protein A conjugated with horseradish peroxidase. And the enzyme oxidizes TMB, and after acidification of reaction solution, I get colorimetrically measurable signal. So now let me tell you how to interpret the signal change. So for unmodified tRNA, the signal is high, and when there is methyl at G9, it goes down and reaches the background level. So this significant difference between signal coming from positive and negative control means that FabF1 is actually an excellent probe for detecting methyl groups, which is hard to do without mass spectroscopy, for example. So this allows me for studying the methylation catalyzed by methyltransferase by watching the conversion of unmodified form into methylated one. So as the next experiment, I have studied methyltransferase activity in time, and I observed that the signal decays in time with decreasing fraction of unmodified tRNA, or the other way around, with growing fraction of methylated form. So here came up a concept of high-throughput screening assay for time-dependent monitoring of methylation, which, unlike conventional methods, wouldn't rely on HPLC, MS, or radiolabeling. And as a proof of concept experiment, I used the FabF1 as an inhibitor towards methyltransferase, since it's a small molecule and it displays affinity towards tRNA, and it worked. So the methyltransferase activity is inhibited. So that's so simple. So, and what's more, this assay, I can detect this activity change with our assay. So that means it can be adapted to a high-throughput platform and serve as a tool for screening activators or inhibitors of methyltransferase. And now, summing up my talk, I would like to emphasize again the importance of mitochondrial tRNAs, and that studying their structure, biogenesis, and function is crucial in understanding the background of mitochondrial diseases. What's more, we have obtained several Fabs that bind to mitochondrial tRNA leucine, and specifically FabF1, the closest to our hearts, displays specificity for unmodified form with 16 nanomolar affinity. What's more, we have used this Fab in ELISA-based assay as a probe for detecting methyl groups, and this platform can serve as a tool for characterizing tRNA modifying enzymes and for screening potential inhibitors or activators of them. And a very happy end of this story. Thank you, Joe, for being such an outstanding mentor, and I'm serious. Thank you. Okay, Yami, you're my best teacher, and thank you for all your kindness and patience. Also, thank you, Tony. I'm so happy to be here, and it's all thanks to you. So thank you for giving me this chance. Our collaborators, Professor Yamin Ho from Thomas Jefferson University in Philadelphia and Professor Tao Pan, thank you for sharing enzyme samples. And last but not least, my dream team. Thank you, Sandeep. I don't know if he's there. I think he's not. Oh, he's here. So thank you for providing your expertise in phage display. Also, Nansheng, thank you for synthesizing 18-mer for splint ligation. Also, thank you, Ben, Deepak, Sorja, Daniel, David, Christina, Simone, Stormy, and Saish. Thank you guys for sharing your knowledge, experience, and I feel so lucky that I can work with you. Thank you. Okay, I will be very happy to answer your questions now. Yeah.
So was the FAB, the F1 binding with cooperativity? It seemed like it was down at nothing, and then all of a sudden it was binding in your gels. So.
Oh, you mean in this?
Yeah, all the way back.
Let me go back.
Methylated
Oh,
versus
this-
non-methylated. Yeah,
this slide.
Yeah.
Yeah.
Yeah. So all of a sudden it really goes up to almost 100% binding. Is there
Yeah.
any sort of cooperativity here?
Again, what?
Is there cooperativity in binding that you're seeing here?
I don't know.
Yeah.
It's not.
I think the concentration ranges are too spread out for that gel, but in more careful analysis, she didn't see.
I see. Okay.
So you have Fab H7 here. So
Oh,
I'm
yeah.
just thinking whether it'd be possible to do kind of a affinity maturation on H7 so that it will be specific for the modified form rather than, and so that you have fabs of one with and one without.
It is. I believe it is possible. So I just didn't spend time for much on this fab, but indeed, on answer results we can see that it's specific for methylated form. So probably, so I have like summer for it, so maybe I can try it.
Okay.
Yeah.
Your job is to make Joe famous, so.
Uphill battle.
Yeah.
Okay. Anyone else?
Great, thanks very much. We have a guest introducer here.
I'm super excited to visit Chicago again after a very short, absence. But it's a great pleasure to introduce the next speaker. I've been working with Błażej Skrobek for 9 months, 10 months, 9 months, something about that. He came to the lab in July. He comes from the Intercollegiate University in Gdańsk on the north side of Poland. And he came to the COS lab in a very enthusiastic and entertaining also time, very intriguing time for us because we just developed this new method and he decided to work with memory proteins together with me. So Błażej is a very special person and I mean it. He's an amazing student. He learns very quickly. He's super meticulous about how he does his biochemical analysis, but above all, he's independent. He learns fast, he's enthusiastic, and he cares about his science a lot. I have never met a student like that before. I'm super proud of him to stand introducing him today. And especially I feel sad because I left him of February, so I treat this as a small surprise and a reward for him so I can introduce him. And it also shows me that even after I was gone, he essentially was able to spread his wings when I was not longer present in the lab to continue this fascinating research. And I was surprised about his result when he was sending me to San Francisco, like, wow, wow. So he has a pretty cool data. I'm not going to spoil anything about his presentation. I just wanted to let you know, guys, I'm really happy to introduce him. And without further ado, Błażej, take it away.
Okay, thank you very much, Paweł, for introducing me. And it is an awesome surprise that you came. And you came yesterday, but you kept it in secret until today, and I was really surprised. And thank you for being my mentor, because I couldn't imagine a better mentor than you. And thank you for teaching me everything, because this is the beginning of my journey in structural biology. And today I am going to tell you about our structural studies on magnesium ion channel core A. And in our laboratory, the main tool for structural biology are synthetic antibodies, or SAPs. They are based on FAB antibody fragments, and we use them as conformational probes to trap our proteins in specific conformation, and as structural chaperones to facilitate crystal lattice formation. And our current goal is to solve the high-resolution structure of the open conformational state of core A. And why is it so important? Because core A is important ion channel in bacteria and archaea, and is critical for the life of these organisms. And it conducts magnesium into the cell. And our core A is from Dermatoga maritima. And what is the problem? Why it is so difficult? Here is the closed structure, which was solved in 2005. And this is a symmetric state and stable state. But in absence of magnesium, there is a problem, because as it was shown 11 years later by cryo-EM, this structure is asymmetric, and it causes its high flexibility. And that's why it poses a big problem for crystallization. And here we come with the synthetic antibodies, which we generate in phage display. And what is interesting about our phage display, we use the technology developed by Paweł, the nanodisk technology. We incorporate our protein in discoidal particles composed of phospholipid bilayer and membrane scaffold protein, MSP, via which we can immobilize this structure without the necessity to chemically modify our membrane protein that can, for example, destabilize it. And the elution from the nanodisk is very easy, because we use only 1% detergent, phospholipid 12. And as you all here know, the phage display requires the library of phages with our subs exposed on their surface. We incubate the immobilized target in nanodisk with this library. Then we perform washing to eliminate non-specific binders, elution, and amplification and E. coli. And the cycle is repeated by 5 times with decreasing target concentration. And after that, we perform validation using different ELISA variants. And what about our target? Our target incorporates into nanodiscs perfectly. Here is the gel from nanodiscs purification, where core A is his-stacked and is immobilized on nickel NTA resin. And comparing between core A loaded nanodiscs and empty nanodiscs, we can say that our nanodiscs are loaded properly. And here in elution fraction, we see our target, core A, and MSP from nanodiscs. And here is the second confirmation that our nanodiscs are loaded. The size exclusion chromatography, elution volume shift. And here is a beautiful monodisperse peak showing core A loaded nanodiscs. And before I will tell you about our current strategy, I would like to introduce you previously generated sub-C12 that traps core A in its open conformational state, because it binds to the magnesium binding site. And that's why even in magnesium environment, when the channel is closed, it is still able to open it. And initially, it was shown at Patch Club experiment that this sub opens the channel, where the oocyte was isolated from the frog. The core A was expressed in this oocyte, and the voltage clamp was attached. And firstly, the control was performed with Core alone, and in non-ionic environments, there is no ion current. And then when we add magnesium ions, there is a big increase in ion current, and then after approximately 1 minute, there is fast decrease because the magnesium ion sensor becomes saturated with magnesium ions and the channel closes. But when the sub C12 was injected into the oocyte, we've got constant ion current until the environment becomes ionless. But what is the problem? Sub C12 showed to be not enough for obtaining crystals of high enough resolution. That's why we performed phage display with epitope masking to obtain a different sub that will bind to the different epitope than C12, and together with C12 will stabilize core A in its open conformation. And during the whole phage display, sub C12 was present in excess and was masking its own epitope. And here is the initial ELISA done after phage display, where the target is in nanodiscs. And it is just to show that we generated multiple potential binders. And here is the more important ELISA done in detergent that is requirement for our crystallization. And comparing between EDTA environment where wild type Core A is in open state, and between magnesium environment where the Core A is in closed state, we can judge if we found any sub that is conformational specific, as for example our control C18, which was generated previously, that binds only in to open state and doesn't bind in magnesium environment, but unfortunately failed in crystallization. That's why we need the other one. And amongst all of them, we didn't obtain any conformational specific. That's why we chose this one, C100, that is versatile and showed the highest expression amongst the others. And we believe that it will be good for as a surrogate for C12 in stabilization of Core A. And before crystallization trials, to be more sure that C100 will bind together with C12, we performed epitope mapping. This is multi-step ELISA, and in the first step, immobilized target Core A is incubated with sub in protein version. In the second step, after washes, we incubate the target with sub in phage version, and in the end, only phage is detected by anti-phage monoclonal antibody. And if the box is blue, it means that possibly both subs bind to the same epitope, because as in the first step the epitope was occupied by sub in protein version, it didn't allow phage to bind, and the phage as a result was not detected. And if the box is red, it means that both subs in protein version and in phage version bound, and the phage was detected. And it showed that C100 and C12 bound to the target together. And initially, I wanted to check C100 capabilities as a structural chaperone, crystallization chaperone, and I crystallized soluble isolated Core A and terminal domain. Here is the elution volume shift that confirms complex formation, and here are the initial crystals from high throughput screening, and one of them gave diffraction at 8 angstroms, and I found it interesting for further optimization. And here are beautiful optimized crystals which gave this diffraction pattern and structure at 2.6 angstroms. Here is the crystal lattice, and this crystal lattice shows the power of our synthetic antibodies as a crystallization chaperones, because most of the contacts are done by subs. And here is the asymmetric unit composed of 2 N-terminal domains and 2 subs C100. And what is the most important, this structure proved that in comparison with previously obtained structure with sub C12, but C100 binds to the different epitope on core A and terminal domain than sub C12. And as shown on this hypothetical model with the full-length protein, both subs can possibly be used in the next step for crystallization of full-length core A. And this step is now in progress. I already have some crystals, but before visiting Argon, I don't know what is inside them, so I cannot tell you if it is successful or not, but I hope yes. And to conclude, subs generated to membrane proteins that are embedded in lipid nanodiscs are much better than using, for example, detergent-based methods, liposomes. These subs can be used as conformational probes and as crystallization chaperones. And what is the most important, sub C12 and C100 can be the next milestone on our way to accomplish this project and obtain the high-resolution structure of open conformational state of core A. And special thanks to Tony, who took me to his lab and who is always kind and gives me a great advice. And thank you also, Tony, for everyday smile on your face. and you always fill me up with positive energy when we see each other. And special thanks to Pavel Dominik, my mentor, who taught me everything and who is always a perfect friend. And it doesn't matter then that we are now far away on the second of America. We are still keeping in touch and still doing the project. Thank you to Ijun Kim, who helped with the initial steps of this project, to Bharat Reddy, who helps me now with crystallization trials and data collection, to Eduardo Perozo, in whose lab I'm doing a lot of experiments, and to Zygmunt de Reventa, who together with Tony Kostiakow and Polish-American Fulbright Commission, they fulfilled my dream about America and science. Thank you to the members of Kostiakow Lab for their help and kindness, to members of Perozo Lab, and to others, especially to Patricia Guzik, who spent a lot of long evenings waiting for me until I will have my complex ready to let me into the mosquito robots, which is closed and locked. And thank you to our funding sources, and thank you to all friends all around the world who are always supporting me. Thank you.
So, is there anything new about the structure of the soluble domain itself? Anything in a different confirmation? I understand that you're probably expecting things to be in the pentameric complex. That's where all the action's going to be. But are there any clues to anything new going on there?
We are still working on the structure because the CDRs have not been built yet.
Ah, okay.
So this information will be released soon.
Okay.
So what happens if you expose bacteria to this SAB? Do they open up and let a flood of magnesium in and die, or what happens?
I haven't checked it.
So it's actually very interesting because we have some problems with expressing some of these antibodies, but not this particular ones, but the ones that are exclusively specific for open state. And the C12 too. Some of them, even though that they're expressing very well on phage, they don't express well in bacteria. So that's one of the hypotheses that they cross-react with the native protein in bacteria.
But the part that it binds to is on the inside of the cell or the
Yes,
outside? Oh, okay. So
yeah.
then
So
nothing
there
would
is
happen,
not that
probably.
much time that you can see each other, but there might be still cross-reactive. E. coli has this protein at a slightly different version, so.
Yes, because this is That's why this protein is important, because this is present amongst all bacteria and archaea. That's why this is a good model for the studies of the general magnesium, the way of transportation of magnesium into the cell.
So when you mix C12, which you have to, which will have a different stoichiometry, right, than C100, you get
Yes. In case of
something that looks like it has 6 fabs on it, or how many fabs?
yes, in case of C12, it binds with a stoichiometry 1 sub per pentamer, and in case of sub C100, I do not have a confirmed data, but elution volume shift suggests that it binds in a bigger stoichiometry than sub C12, so it can be 3, 4, 5.
So it's a broad peak that you get, or?
It is not a dispersed peak, but judging on the only shift, I cannot say how many exactly. That's why for crystallization trials, I'm using, I firstly incubate the protein with 1 to 5 molar ratio of sub C12, and then I add big excess of sub C100s to make sure that all other epitopes are occupied by this sub.
More questions? Well, we have a break built in here, but since we're a little bit running ahead of schedule, why don't we have 15 minutes and then come back here a little bit early and continue on, because after this we're going to have our annual Polish supper, so with wine and drinks and a selection of Polish food, so we can get to that earlier if we finish it. Okay. So 15 minutes, which will be when we come back at 20 of